Welcome back, everyone, after the break. And now we move on to our third speaker of the day today, Dr. Jitu Patel. He is a lead scientist at the Environmental Microbial and Food Safety Laboratory at the US Department of Agriculture since 2003. His research interests include pre-harvest contamination of fresh produce, bacterial attachment and biofilm formation, natural antimicrobials, and alternate farming practices. Prior to joining the USDA, Dr. Patel was technical director at Tetley USA Incorporated, where he was responsible for quality assurance, product development, and regulatory compliance programs. He served as a senior scientist at Future Medical Technologies Incorporated between 1995 and 97, where he managed research on diagnostic kits for the detection of pathogens in food as well as clinical samples. Dr. Patel received his Bachelor of Science and Master of Science in Dairy Science from Anand Agricultural University in India, and a PhD in food science from the University of Georgia in the US in 1994. And I have known Dr. Patel since 1994 when I was a graduate student at the University of Georgia in Athens, Georgia. And I have collaborated with him on several food safety projects. He's been doing a lot of interesting research, and he accepted my invitation to give a talk to us today, even though he's on Eastern time. So, it's a bit late for him. So I really thank Dr. Patel for talking to us today. His talk will be on pre-harvest treatments to control bacterial pathogens on fresh produce. So it's all yours, Dr. Patel. Thank you.
Hi, good afternoon, everyone. Thanks to University of Arizona Food Safety Consortium and Dr. Ravi Shankar for inviting me and present this particular research, which we have conducted at USDA Beltsville, as well as our other facility, contractor facility in Pennsylvania. As we all know that fresh produce can be contaminated at the farm level all the way to the fork at the retail level too. And many of the factors that could contaminate at the farm are like egg water, compost, we also call as biological soil amendments, animal vectors, farm equipments, workers, and so forth. And if there's a contamination at the farm level, there's a significant risk of cross contamination when the product is introduced into a processing system. So it is very important to control contamination at the farm. Now, one of those huge factors responsible, which is egg water, and scientists at University of Arizona like Shanna Rock and Garba and Sadhana and all, you guys have been doing wonderful research on egg water, and studies have been conducted in the field level too, and those research was presented at the CPS meeting this year. Not only egg water, but also the treatment options. So I would cover some of the different aspect. What we would try to cover today is a potential antimicrobials that could be used on fresh produce. Just the produce is about to be ready to harvest. So before we started doing research, we wanted to see what happens on different surrogates that we could use in the field study. So in one of these field study, we used 3 different lettuce cultivars, Annapolis, Salinet, and Coastline. And we used 2 different surrogates. One is E. coli on 57H12. This strain was isolated from a creek water, and the creek is running through just nearby the farm that most of our research has been done. And second strain we used was a K12. And to make research a little bit interesting, we used three different kind of water: groundwater, roof harvest water, which is RW, and untreated wastewater. With H12 strain, on day zero, we were able to recover around 4 log CFU, irrespective of the cultivars on Annapolis, lettuce, salinate, or a coastline. And as expected, we found die off of this H12, and at the day 10, we didn't see any difference because of kind of water we use for the irrigation. And the similar results with the K12, which is a lab strain. What we observed is here, the recovery of this particular surrogate was lower. It was around 3 log CFU compared to 4 log CFU you obtained with the H12. Similar die-off pattern observed on K12 strain also. And if you look at around 10 days, the numbers were not significantly different in most cases. And in some cases like coastline and a salinate, type of water used for irrigation, it did not have any effect at all, very close to 1 log CFU per gram of spinach reported in salinate and coastline, irrespective of uh, water used for irrigation. So we did some images of a lettuce leaf, and Annapolis, which was a red lettuce, and we looked at the stomas and structure, and see, this is Annapolis has more wavy structure compared to coastline, which is more waxier surface, and the salinate is also a more wavier structure compared to coastline. So there were differences in the leaf characteristics. And then we also checked for stoma density. And stoma density was significantly higher on Annapolis, close to about 100 stomata per millimeter square compared to salinate and coastline. But our results did not match with this particular stoma number, and we can see from those results what we found, that E. coli H12 seemed to be a more suitable surrogate for the study because one is it was a natural habitat. Um, it persisted at the higher level compared to K12 strain, and when the treated water was used, the recovery was the lowest on all these lettuces. As expected with most bacteria study in the farm level as well as the growth chamber level, we have observed the die-off rate, but the cultivar affects the die-off rate. So the lower recovery of E. coli was observed Annapolis lettuce on day two day post inoculation. So this is a little bit an awkward from the previous slide. We saw that Annapolis had high number of stoma, and it might have more bacteria attached to it, but this is unusual. So what we think that besides the stoma density, the waxy surface, the cells leaves roughness and waviness and other factors could influence the persistence of bacteria on the leaf. And we also did some this secondary water and roof harvest water for indicator bacteria, which we haven't shown here, especially with total and fecal coliform, as well as generic E. coli. And we didn't find any difference, but mind well, there's the bacterial quality of roof harvest water and secondary treated water was as good as groundwater. So if these two waters had a higher number of generic E. coli or coliform, the results could have different. So let's talk about pre-harvest interventions. So um, we have studied different interventions and we'll go through those. In a biocontrols, we used lactic acid bacteria. Now, lactic acid bacteria include lactobacilli, which are very commonly used in a yogurt and fermented milks. We have some bifidobacteria, which provides the immunity. We have Pediococcus, Propionibacterium, which is used in a cheese. So it could be a big group of lactic acid bacteria. Those bacteria have been proven to be again and bactericidal, and they have been used in to prevent the growth of spoilage bacteria as well as some pathogens in the food. The bacteriophages are like natural killer system, and we have commercially available bacteriophages like Salmophrase and Lacticil and all. Those Listosil, sorry, or Ecosil. Those are commercially available, and they have a specific application on certain foods. And we also have predator bacteria. Predator bacteria, basically they engulf the target bacteria, and two of the common predator bacteria studied are myxobacteria and Delovibrio-like bacteria. Those bacteria have been evaluated to reduce soil-borne pathogens, and we also try to do some research on bacterial pathogens. Besides biocontrol, we evaluated some antimicrobials too. The one of those antimicrobial benzyl isothiocyanate, we call BIT, which is commonly occurring in brassica vegetables. Brassica vegetables have glucosinolates compounds, and that compounds, when are hydrolyzed by tyrosinase, this BIT is released. And BIT has a very, not besides antibacterial, it has also therapeutic properties. Caprylic acid is one of the component in a coconut milk, and that is we have studied post-harvest and found significant reduction at very low concentration. It has been also used to kill bacterial pathogens in a water. And octanidyne dihydrochloride, OH, is a chemical. It is non-toxic. It is not absorbed by the skin. In fact, it has been used widely in UK and other European countries as an ingredient in a toothpaste. It has been used in a shampoo to kill MRSA, and it's also used widely in the hospital to control contamination. So those biocontrols and those antimicrobials we have studied in the lab. So first, one of the first study we had was biocontrol of Listeria on spinach with the lactic acid bacteria. We had 4 different spinach cultivars, and those cultivars were selected by local growers in Mid-Atlantic area. This study was conducted in a farm in Pennsylvania, and the growers will decide the cultivar based on the season and then marketability. And we also wanted to look into different leaf pattern. And as you can see here, the abundant is very dark greenish leaves compared to others. And same Matador has different characteristics than Palco and Butterfly. And we had a different plot size where we inoculated a cocktail of Listeria innocua at 5 log CFU per gram. Now, in another study, we found that anytime we do persistent studies on a pre-harvest, um, the bacteria we prepare in fecal slurry. When we prepare bacteria in fecal slurry, we get a better persistence than the bacteria prepared in a lab media. So all inoculum was prepared in a cocktail slurry. And after inoculation of this Listeria innocua cocktail, after about 2 hours, we used lactic acid bacteria spray. Now, these lactic acid bacteria used were Lactobacillus lactis, Plantarum, Johnsoni, and Acidophilus. These strains are commercially available, but our collaborators screened those strains for antibacterial activity in vitro, and we use a cocktail of these 4 strains as our LAB treatment. At day 0, 3, and 5, spinner samples were collected and analyzed on a MOX for innocua population and an MRS to see LAB population. So here, with the abundant, the very different spinach cultivar, what we observed that the day we sample, the day after inoculation and treatment, with the LAB, we found about a little bit more than 1 log reduction of Listeria with the treatment. But the reduction was not evident on day 3 and day 5. And initial attachment was the 4 log CFU on abundant. When we compared this to a butterfly cultivar, and the results are a little bit different. Now look at the stoma density, which is about half, 56 versus 101. And here the results are different. We get a little bit about 1.5 or so log reduction on day 0, and also on day 3, but not day 5. The Metados cultivar attachment was much higher, significantly higher, close to 6 log at day 0 compared to abundant and butterfly. And we had marginal reduction on day 0, and then significant reduction on day 3, and marginal reduction on day 5. And the last one is a Palco, which has the lowest number of stoma. And they, in this particular cultivar, we observed a reduction of Listeria with LAB treatment at day 0, day 3, and day 5. And day 0, we had a significant reduction. So when we look at the all four charts, we can see in abundant, LAB is effective only at day 0, on butterfly on day 0 and 3, and other 2 strains at 0, 3, and 5. And the persistence also varies. It's about 4 log CFU on abundant and butterfly versus close to 6 on other 2. So this could be attributed to, besides stoma, the waxy surface or cell leaf roughness of the leaf could have attributed to differences in their persistence of this Listeria. So after this study, we thought that we can do a similar study on lettuce cultivars. So with the lettuce cultivars, we use 2 different lettuce, green lettuce and the red lettuce, green star and red fire. And during the first week, we can see that there's some reduction, but it's not significant with the green star at day 0, day 3, and day 5. With the red fire lettuce also, LAB treatment has no significant reduction. So this was a little bit surprising to us. With the spinach, we had an instantaneous effect of lactic acid bacteria on listeria, but not on the lettuces. So we thought that can we have a booster dose? I think booster dose is very commonly heard right now. So we gave another dose of LAB on a second week. So this basically is day zero is a 7 day after day zero. And so we give another shot of LAB and the wow, results are totally different. With the green star, we found a significant reduction of listeria on day zero, and not significant, but reasonable reduction with day three. And same thing happened with red fire lettuce. So it seems that this LAB work different on different um, product category as well as cultivars. With the spinach, we were getting results on day zero. Here we have to wait for 7 days, another booster dose to get anti-listeria effect on lettuce. Besides listeria, we also did 0157H12 on these lettuce studies. And here, the persistence, especially with the red fire lettuce, was much higher compared to green star lettuce, and we found significant reduction of 0157H12 by lactic acid bacteria at day zero. And day three and day five, the numbers were not significantly different compared to control. So here is the difference is due to the kind of pathogen. With the listeria, we were not getting a reduction on first week on any of those particular lettuces, but here on first week on day zero, we got significant reduction, more than 2 log or so with lactic acid bacteria on 0157H12. We still continue the second week with as we continue with listeria, and results is totally different with green star lettuce. Actually, on green star lettuce, we don't see any effect of the second dose on 0157H7. But the trend seems to be continued with the red fire lettuce. So it again, besides the pathogen, the kind of lettuce also influence the effect of lactic acid bacteria on E. coli 157H12. Since we got variable results on those green star and red fire lettuce, we thought that why don't we do real pathogens? And so this study was done in the growth chamber. In a growth chamber, we used similar study with real like Listeria monocytogenes and 0157H7. And we found noticeable difference with Listeria monocytogenes. On day zero, we found significant reduction with day zero with lactic acid bacteria. Now, you may see that in the field study on a green star, there was no effect of LAB on Listeria innocua, but we found significant reduction on day zero at day three, and also lower recovery on 0157 and H7. So what it implies that many times we do a study in the lab or in a growth chamber where all other environmental factors are controlled, we get different results than what we see on a field study. And the similar study was continued on new red fire lettuce. Now, compared to green lettuce, this particular red fire lettuce had higher attachment or higher persistence of Listeria monocytogenes, which was 5 log. But again, we can see there is a significant reduction of Listeria monocytogenes, as well as E. coli 157 on day 0. So similar to green lettuce, red fire lettuce too, we found significant reduction on day 0 with both pathogens, which was not that feasible in a field study. Another compound we use is a benzyl isothiocyanate for biocontrol, and for that we used 4 different spinach cultivar, lakeside, butterfly, matador, and regiment. One question may come: why we have different cultivars at different time? And one of the reason is we use different seasons for growing the spinach, and the farm manager will coordinate with local farmers or growers, and based on their recommendation and availability of seeds, we would have different cultivars. And BIT was used because we found BIT is an excellent antimicrobial to kill salmonella on alfalfa seeds. So even though this is a different chemistry, we thought, can we use BIT on a spinach? So protocol was the same. We had 0157H12 strain inoculated on a spinach, and then a BIT was added at 0.5% or in combination with additives, and then they were sampled at 0, 3, and 5 days for surviving populations of H12 strain. This is a little bit cloudy, but if we see at the lakeside, there is no effect of a BIT at all on H12. The numbers are 4 log on day 0. In fact, at the higher, like day 3 and day 5, we found an increase in recovery of H12 when they were treated with the BIT or BIT with additives. Similar results were found with the butterfly. We do not see any effect of BIT at day 0, day 3, or day 5. So the effect is in fact, it supported the growth of H12 on day 3 and day 5. Same thing with the matador. Sorry, day 0, here is an important thing. At day 0, we had about 1.5 log lower recovery with a BIT and BIT and additives at day 0 with the matador cultivar, but later on the effect was not apparent. And regiment also, we had about 1.5 log lower recovery when BIT was used with the additives. But then later on, the effect was not significant. So it seems like BIT and additives may have some promises, and that promise is limited to certain cultivars only, matador and regiment, and not others. In fact, it supported the growth of H12 strain on other cultivars. Now we are going to cantaloupe. And cantaloupe study was conducted in a Bellsville facility here, and we used the cantaloupe which were about to be harvested. We sprayed with Listeria innocua, and this cantaloupe was marked with 2 different sites, about 50 square centimeter, and each area was sprayed with 3 ml of inoculum, and after air drying for about an hour, we sprayed with 3 ml of lactic acid bacteria. Now, lactic acid bacteria used in this study are different. These L. plantarum and pedococci are not commercially available. In fact, they were isolated from canine feces at University of Connecticut, and they have done extensive study in vitro, and they found that these 2 strains were had a good antimicrobial property against common bacterial pathogens. So we use this to strain as a cocktail, and at 0, 5, and 7 days, we sampled the cantaloupe and tested for surviving populations of listeria, and we can see that listeria were significantly reduced on day 3, day 5 and day 7. Um, and LAB population, which were about close to 5.5 log or so, that also reduced a little bit. But even after 7 days, there were LAB populations um, on cantaloupe. So it seems like that LAB cocktail used, proprietary cocktail used in our study could be effective in reducing or killing 2 logs of listeria and 1.5 log by day 5 and 7. These octanoidine dihydrochloride that we talked about earlier as a chemical, we also use that on a cantaloupe, and these cantaloupes were grown in a Fulton farm. And here we did a little bit different. We, instead of spraying on 50 square centimeter, we used a bucket and a bucket of inoculum of Listeria inocui or H12 strain, and whole cantaloupe was dipped for 10 seconds. Once it air dried, the whole cantaloupe was sprayed with the 15 mils of OH at 0.1 or 0.2% concentration, and they were sampled at 0, 14, and 21 days. The reason we had extensive days of sampling, because this study was done a little bit earlier, not as ripe as the previous one, and we wanted to see the long time effect of OH. And the study was conducted in a high tunnel, which is a closed environment, so there would be a lesser effect of sun exposure as well as no rain. And the other study was open field. And in the high tunnel at 0.1% OH, about 3 log reduction of H12 population was observed, and about 5 log reduction of H12 observed at 0.2% OH. While in a field study, the effect of concentration was not significant. Even at 0.1% concentration of OH, we found about 6 log reduction of H12 strain on a cantaloupe at 14 and 21 days. And study with listeria, we had 2 separate trials at 2 successive years with the listeria on a high tunnel. And with the high tunnel, we had about 3 log reduction at 0.1% concentration, and close to 4 to 5 log reduction at 0.2% OH. The next year, when we did the study again, we found similar results at 28 days. Here, there's a difference. On 1 year, we sampled at 21 days. The next year, we extended to 28 days. And we still found anywhere from 3 to 5 log reduction in the high tunnel. This study was also conducted simultaneously in a field setting. And in the field setting, we had a little bit different results compared to high tunnel. For example, here in one of the first year study, control and 0.1 had 0.1% OH had a similar effect, and with the 0.2% OH, we had a significant reduction of listeria on a cantaloupe at 14 and 21 days. And with the field study here, in at 0.1 and 0.2% OH, we found significant reduction with on Listeria on cantaloupe, we were unable to sample at 28 days because most of those cantaloupes were rotten by that time, and it was not enough samples available to do the study. But overall, we can say that um, OH was very potent antimicrobial and able to kill H12 and Listeria innocua both on the farm setting, whether it's a high tunnel study or field study. One of my colleague and her group, she conducted study on Panibacillus. Panibacilli, this particular antagonist was isolated from plants which are native to Virginia Eastern Shore region. And this particular strain, TS15 strain, has shown significant inhibition of bacterial pathogens in vitro, as well as tomato fruit. And what they did is they used in the field study, at attenuated salmonella strain was used, and they spot inoculated this particular strain on leaves and blossoms and fruits. And the inoculum level was about 20 microliter per leaves. And after that, they used this Panibacillus strain, 40 microliter per leaves or per fruit. And what they found is the chart looks a little bit tricky. Is death rate in fold is not a log reduction, but we can say that the first one on the leaves, we had about 12-fold reduction on salmonella with on the leaves and blossoms, but not on tomato fruit. And 90% of the samples, the leaf samples, they had non-detectable level of Salmonella newport. So this is another biocontrol as could be used on Salmonella on tomato plant. And this is not our study. This actually was done at the University of Georgia. They call it Patel sanitizer. The sanitizer is a combination of 0.5% luvulinic acid and 0.05% of SDS. And these sanitizers, they have used in a post-harvest on a romaine lettuce, and they have found significant inhibition of Salmonella and O15787. It did not influence the sensory quality of romaine lettuce, and this was the pre-harvest study they did in two different approaches. One is a treatment approaches in which they inoculated bacteria first on the surface of tomato plant. So this was a more a growth chamber kind of study on a 4 weeks old plant. They sprayed pathogens first, and then after pathogens, they were treated with either water, chlorine, or Phet-L. And the second approach they conducted was a preventive approach, where plants were treated with this water, chlorine, or Phet-L sanitizer, and then contaminated with the target pathogen. And then the different sample plant tomato leaves were analyzed for O157 salmonella and monocytogenes. So when feed treatment was used prior to inoculation, which is a preventive approach, all three pathogens, typhimurium, Aztec, and Listeria monocytogenes, they found significant reduction using Phet-L sanitizer, about 1.3 log reduction in typhimurium, 0.8 log in Aztec, and 1 log reduction of Listeria monocytogenes was achieved as a preventive approach. And the sampling, this is an average of 10 tomato leaves collected from 5 tomato plants, and as the asterisk shows the significant difference. The second approach was a treatment approach, and also in that case, they found significant reduction of Salmonella typhimurium, Aztec, and L. mono using FIT-L. And again, the same results are CFU per square centimeter of 10 tomato leaves collected from 5 plants. This FIT-L treatment was used as a field study. In a field study, they had two separate trials, and first trial they had 100 samples, and the second trial they had 100 samples. And before treatment and after treatment, they did not find any O157, so as salmonella. But listeria, they found 8 samples positive prior to treatment, but after those treatment, all 8 were killed. And those 8 listeria were, 2 of those were Listeria monocytogenes, 4 were Ivanova, and 2 were Grayi. Interestingly, it also reduced aerobic plate count and coliforms in both trials. In first trial, the plate count reduced from 5.8 to 4.1 log CFU per square centimeter, and about 1.2 log reduction in coliform. So I'm winding up my last slide, and this is a in summary, what I would say that lactic acid bacteria as well as other antimicrobials can be used potentially for controlling contamination of prior to harvest. But these antimicrobials, their effect varies. Some may require longer days to be effective, and so in some cases it would be day zero. In some cases it would be longer. We also found that field studies are important because as on the growth chamber, we got significant reduction of pathogens, but the results cannot be duplicated in field studies. And we use multiple lettuce cultivars and spinach cultivars with different leaf characteristics, and we observe different results. So that is very important, that it's not a one size fit all approach. We may have to use different chemical, different concentrations depending on the cultivars. And second thing is this approach could be more suitable for the smaller farm because large volumes of prey would be required. And other parameters were also important, like for example, sensory quality. Caprylic acid was an excellent antimicrobial in the post-harvest, but then it was leaving some residues on the leaf, and that affected the sensory quality of leaf. Second thing is the cost. OH kills like anywhere from 4 to 6 log CFU on a cantaloupe, so it's very good antimicrobial to use, but that's quite expensive. Second thing, we don't know about the effect on environment of residual chemicals on soils and rhizospheres, and the microbiome. A study conducted by one of our group scientists, they found some changes in microbiome. Major taxa remain the same, but those are the factors we need to consider those. And last, I would like to thank all my team, Dimitru, Shiva, Amy, Paul, and they all conducted this research and contributed one or other way. With this, I will end, and I would entertain any questions if you have. Thanks.